Good morning, good morning, good morning. Hello. Hi, kids. Hi, students. Hi, everyone. Good morning. So, hi, everyone. Welcome to Unacademy NEET English. Am I perfectly audible and visible? Hi, good morning, good morning, good morning. So, how are you people feeling? In the morning, we are having a session, right? It's a very good time for the revision as well. So, that is why I decided that we should keep a class early in the morning. And obviously, it's not that early. Hannah, it's not that early. So, kids, is that okay if I'll keep your session at 6 a.m., 7 a.m., or 8 a.m.? Thank you so much, bache. Huh? Is that okay if we'll keep your session at 6 a.m., 7 a.m., and 8 a.m.? Huh? Seriously? Even it is okay. For me, it is not. For me, it is not. I cannot wake up that early. I literally cannot. So voice is fine. Everything is fine. Yes. So you know that it is the Ace the Race series in which we have started your class 12th biology syllabus, and no doubt initially we are going to move little slow, but soon we will again come to the marathon part, right? I was just waiting for student support that are they okay in taking the classes in the morning, or otherwise we would have made some changes. But I think the slot is perfectly fine. We will continue it and we will finish the class 12 syllabus in the morning and class 11 syllabus in the evening, right? So guys, you can see our channel here. First of all, I would like to say thanks to all those students who mentioned their comments here. Thank you so much, Hemrish. Thank you so much, Jennifer, Saravan Vijay, right, Abhishek. Okay, thank you so much, Abhishek. It's a lovely comment. Shri Harsha, thank you, thank you, thank you so much, Shri Harsha. I will take care of it. And Shweta and Krishna, thank you so much. Thank you. So, guys, it will hardly take your 10 seconds to write a comment. So, it's a humble request. Please, 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 and please, in the comment section, do let us know you like the session or not. Whatever is there in your head, if you have any suggestion, please put it in the comment section, right? Right, it will literally take hardly your 10 seconds. And moreover, now you guys can see our channel here, the Unacademy NEET English Kids. So see, you will get everything on this channel, everything. See, whatever updates are required for your NEET examination, you are going to get it here. You can see the video section, bache, right? Shreyas sir has updated something, Wazeem sir, me, HSP sir, right? From time to time, we will provide you all the essential, all the important information. Here in this video section, you guys can see, bache, about the CMC, Vellore, AFMC, AIMS, and even cutoff of the South for the South Indian colleges. Everything, we used to put here. So stay tuned to our channel, and if you are new to our channel, do subscribe our channel. And moreover, kids, if you want to be the part of any of our batch, right, you can find the link in the description box. But that's a request. Please join the batch ASAP, because otherwise a lot of backlogs will be there. Okay. So see here, right, today also, not today, I think, yes, your Wazeem sir is going to take the marathon, right? I will take the marathon. So you can see our schedule here. Okay, so don't worry, we will finish your syllabus before NEET 2024. You have to be consistent, you have to follow our channel, and you have to practice the questions. Okay, this is what you people need to do. Okay, ma'am, screen is tilted. Yeah, even I can see. So guys, is it manageable? Just a minute. Is it manageable? I think it is. Now better or not? I don't know what happened all of a sudden, but I think it's fine now, right? It's fine now. Okay. Okay. It's manageable, right? So after the class, I'll rectify it. I hope it's manageable for you, right, bache? So you can see, you can just click on any video. What you have to do? You can see the description box here, and here is the link for the batch. Just click here, fill your details, and you can be the part of our batches. Okay, you guys can join our batches. Clear, bache? Okay, so let's continue with this chapter, human reproduction. Last time we were, we started male reproductive system. Today, this is what we are going to complete. So let me know in the chat section which one is the primary sex organ in the case of males. Right? Let me know in the chat section the primary sex organ in the case of males. Quickly. Which one is the primary sex organ in the case of males? Yes, kids? Yes. And why do we call testes as the primary sex organ? Why? Why testes are considered as the primary sex organ? The reason is there will be the spermatogenesis in the testis, first point. Secondly, they are going to release the male sex hormone, that is male and androgens, testosterone and dihydrotestosterone. Right? So why do we say that in the case of female ovaries, in the case of male pair of testes, they are the primary sex organs? Because in these sex organs, there will be the gametogenesis, and then they are going to produce the hormones, the sex hormones. Clear, bache? This is what they are going to do. Okay, they will produce what? They are going to produce the sex hormones. Right? That is the reason. First point. Now, let's revise this again. So in the last class, we have already completed the scrotum. So you know that scrotum is a pouch-like structure in which pair of testes are present. What is a scrotum? Scrotum is a pouch-like structure in which pair of testes are present. Yes or no, bache? Yes or no? And now, here in this pouch-like structure, it is going to regulate the temperature. What will it do? It regulates temperature. It's a thermoregulator. What is it, bache? It's a thermoregulator. Clear? Clear? So in the case of males, you know that testes during the 7th month of pregnancy or after the 7th month of pregnancy, initially testes are abdominal in the case of males. Initially testes are what? They are abdominal in the case of males, but later on after the 7th month of pregnancy, they will descend into the scrotum. Right, bache? They will descend into the scrotum, and you know that with the help of what? With the help of inguinal canal. With the help of what, bache? With the help of inguinal canal. This is what we have covered in the last class. Yes, bache? So if testes, they are not descended into the scrotum, there is a condition that is cryptorchidism. What is the name of that condition? The condition is cryptorchidism. Clear, bache? Clear, bache? And this inguinal canal is also a connection in between this pelvic cavity and the abdominal cavity. Students, if you are attending this lecture for the first time, we have already covered all these points in the last class. So that is why today quickly we are revising all these things. Okay, so today it's a quick revision session. Are you getting my point? Today it's a quick revision session. Fine? Fine. So that is the cryptorchidism. Now here in this inguinal canal, what will you see? You will see spermatic cord. Isn't it? What will you see? You will see spermatic cord, right? This spermatic cord will have gonadal arteries. It will have gonadal veins, the nerves, right? Right? Even some muscles are also present there. The connective tissue will be there. The connective tissue will be there, right? That's what we have covered. Now, as I said, this scrotum is a thermoregulator. It will regulate the temperature. It will maintain the temperature here less than 2 to 2.5 degrees Celsius than the human body, right? Right? So basically, if human body is having a temperature of 37 degrees Celsius, and the scrotum, the temperature will be near about 35 degrees Celsius. Okay, it will be near about what? It will be near about 35 degrees Celsius. This is what you people need to keep in your head. Okay, okay, that is the one point. Now, the second thing here is, right? Just have a look. The second thing here is, right? I told you about the muscles. Do you remember? Cremaster muscle and dartus muscle. Cremaster muscle and dartus muscle, they help in positioning the testis. They help in positioning the scrotum. Like if you talk about the dartus muscle, right? Dartus muscle is present here. It is lining the scrotum. It is lining the scrotum. And when you talk about the cremaster muscle, even it is present in the wall of scrotum and even in the inguinal canal, right? Inguinal canal. Guys, I'm repeating it again. It is basically a revision, right? We have already covered it in the last class. So it is what? It is basically the revision. What is it? It is basically the revision. So up to this part all clear? Yes, everyone. Up to this part all clear? Sure, bache? Are you sure about it? Very good. Now, what is the next point here? So dartus muscles. What will they do? They will contract. They will contract in cold weather and relax in warm weather. This is what you need to keep in your head. Right, they will contract in cold weather and they will relax in warm weather. So when they will contract, obviously, imagine this is present in this is lining the scrotal wall. So it will contract. Obviously, the skin of the scrotum will be wrinkled, right? It will be like, you know, some kind of aggregates will form. It will be wrinkled, right? And when you talk about the cremaster muscle, which is present in the wall of the scrotum lining, even in the inguinal canal, it helps in the elevation of testis, right? It helps in positioning and elevation of testis. So when the conditions, right, when the conditions outside, or you can say that when the temperature is low, the testes will be pulled towards the pelvic cavity. They will absorb the heat from that pelvic cavity, right? And when they will, you know, when the temperature came down, again it will came down. Okay, okay, mera bacha. Okay, is that clear? Is that clear? So that is what we have already covered in the last class. Then we were talking about the testis. Okay, so kids, quickly in the chat section, please tell me about the coverings. Now we are going to talk about the testis, which is present in the scrotum, right? The pair of testes which is present in the scrotum. So quickly tell me about the coverings in a sequence. In a sequence, just let me know about the coverings. Quick. Everyone just be quick. Yes, exactly. So when it comes to the covering, what do we have, bache? Right? There is one trick, that is wab. There is one trick, that is wab. Right, that is wab. So for the covering, bache, the word is tunica. So it's wab, V-A-V. So it's tunica, outermost is tunica vaginalis, middle one is tunica albuginea, and innermost is tunica vasculosa. Right, innermost is tunica vasculosa. So outer one is vaginalis, middle one is albuginea, innermost is vasculosa. Right, bache? So outer vaginalis, totally, it is covering the testis completely. Okay, then comes tunica albuginea. It will also cover, but posteriorly there will be a kind of space, mediastinum. But okay, we don't need that, right? We don't need that in detail, right? Right? Clear, bache? Clear? And innermost is the tunica vasculosa. Komal, bache, your question can come from this part. So please focus here. Tunica vasculosa, innermost covering, and this is going to cover the testis internally. It is also covering these testicular lobules. Fine. It is also covering these testicular lobules. Is that clear? Is that clear, bache? Yes, everyone. Tell me. Is that clear? Now, I have already given you the example of the building. I'm repeating this example again. Let's say there is a building having 250 floors, right? And each floor is having 1 to 3 flats, right? Or apartments, whatever you want to say. 1 to 3 flats or apartments. So same is the case here. Now in the testis, right? Within the testis, you will find some compartments, and for such compartments, we use the word testicular lobules, right? For such compartments, we use the word testicular lobules. Is that clear? Right? So they are 250 in number, and in each testicular lobule, bache, right? And in each testicular lobule here, you will find 1 to 3 highly coiled seminiferous tubule, right? In each testicular lobule, you will find 1 to 3 highly coiled seminiferous tubule. This is something very important. Fine. This is something very important, and focus on this word. They are highly coiled. Clear, bache? They are highly coiled. Clear bache? Clear bache? So if there is any doubt in this part, do let me know. And see bache, I know, you know, I make these diagrams, some rough diagrams, but trust me, these diagrams are very good to explain a topic, right? Why? These diagrams are literally good if we want to explain a topic to you. So just have a look and understand the topic, because ultimately we need to revise what? We need to revise the NCERT. Right? What we need to do, bache? We need to revise the NCERT. Clear, bache? Clear, bache? So now here you all can see that these seminiferous tubules are present in testicular lobules. Yes or no, Shri Harsha? These seminiferous tubules are present in these testicular lobules. Now, what will they do? Yes, what will they do? All these seminiferous tubules, they will join. They will join. Like, let's say one line, one pipe, second pipe, third pipe, fourth pipe, all the five pipes together are making, they all join together and they are making one large pipe. Or you can say that all the pipes, they are opening in one tank, like this, right? Like this you have to keep in your head. Okay, so these seminiferous tubule, all these seminiferous tubule, what will they do? Here they are going to form a network, right? Here they are going to form a network of tubules. Although there are some other words also, but we will follow the NCERT here, and you will get the rete testis, right? Rete testis here. That is what we were discussing in the last class. Clear, bache? So here the seminiferous tubules, they all are going to form a kind of network, right? So that network is what? That network is what? That network is known as rete testis. That network is known as rete testis. Everyone just write it down. For Kana, long time, bache. Just write it down in the chat section quickly. This network is what, bache? This network is rete testis. Now I think screen is not tilted. Now I think it's fine. Yes, kids? Is everything okay with the screen now? I think now it's not looking tilted or something. If it is, do let me know in the chat section. And yes, we have so many students and number of likes are quite less. So like the video right now. Just like the video right now. If it is still tilted, just let me know, bache. If it is not, do mention it in the chat section. Sure? Okay. So now from this rete testis, some other, you know, ductules, they will arise. Now, from this rete testis, right? From this rete testis, right? We will also find some ductules. So you can say that this is a network, right? This is a network basically. What is it, kids? But kids, this is a network. So from this network, you can say this network fuse to form ductule, right? To form ductule, and that ductules are what? There are 15 to 20 ductules, and that is what? That is vas efferentia or vas efferens, right? You can also call it as vasa efferentia. You can also call it as vas efferens. So the sequence is important, right? Right? The sequence is important. Let's say here you will see that rete testis, they all are fused and they have formed what? They have formed what? They have formed these 15 to 20, near about 15 to 20 ductules. We are calling it as vasa efferentia. We are calling it as vasa efferentia. Now next to it, all these vasa efferentia, right? They will join, or they will, you can say that, open up in what? Right? All of them, they will form what? They will form your epididymis, right? They will form your epididymis. Is that clear? What will they form? They will form your epididymis. Clear, bache? Clear, bache? Yes. So this sequence is important. I'm repeating it again. This sequence is important. It's very important. Epididymis, right? Epididymis. Clear? So it is the place where there is the temporary storage and maturation of sperms, right? What is epididymis? It is a P by Q as well, bache. What is epididymis? It's a structure, it's a place, it's a duct where there is the temporary storage and maturation of sperms. Temporary storage and maturation of sperms. Temporary storage and maturation of sperms. Are you getting it? Right? And let me tell you, students, sperms can get stored there for months. Okay. sperms can get stored there for months. Are you getting it? For months they can get stored there. Okay? Clear? Clear, clear, clear? Done? So this is the epididymis. So this sequence is literally important. The sequence is literally important. Okay? So now let me show you one diagram. See, this is the diagram. Testis, testicular lobules having 1 to 3 highly coiled seminiferous tubule. They all are forming a network. We are calling it as retitestis, right? We are calling it as retitestis. Now, from these retitestis, what is arising? These vasa efferentia, and they all will open up here in this part, right? So this is what? This is epididymis, right? So you can say that up on the upper side of the testis or on the dorsal or the posterior side of the testis is having this epididymis. Clear, bache? Clear, bache? It is the epididymis. So now if you look at this epididymis, further it is forming a muscular duct, and we are calling it as what? We are calling it as, yes, bache, we are calling it as vas deferens, right? Vas deferens, and even the ductus deferens. Vas deferens and even the ductus deferens. Is that clear? Vas deferens and even the ductus deferens. Clear, bache? Are you sure? Are you sure? Yes? 110%? 110%. So now this vas deferens or ductus deferens, it will move ahead. So basically how these ducts will pass further, all these ducts, they are forming vas deferens ultimately, which will move to the abdominal cavity. It will coil over the urinary bladder, right? Right? So we will be having the pair of this vas deferens or ductus deferens, and it will coil over near to the ureter of the kidneys. It will, it will go there, it will coil over there and over the urinary bladder, and then finally it will enter. Okay, so that's the point. Any doubt, kids? Any doubts? Now what we need to focus? We need to focus on this part, that is the seminiferous tubule. Okay, let's go one by one. Let's go one by one. So ultimately from this part, what type of question can come in your examination? First of all, let me tell you this, and then bache, okay, then I'll tell you the next question. So ultimately, what will be the root of the sperm? Firstly, the seminiferous tubule, where it will form, right? Seminiferous tubule, where it will form, and then from the seminiferous tubule, what will be there? Retitestis, right? From retitestis, there will be the vasa efferentia, right? There will be the vasa efferentia. Then there will be the epididymis. Then there will be the vas deferens. Clear, bache? And this vas deferens will further receive the seminal vesicle, and you will get what? There will be the ejaculatory duct, and finally it will open up there in the urethra, right? Urethra. It will open up there in the urethra, and then finally the external opening. Basically this is the route, this is the pathway. Yes or no? Yes or no? Clear? Clear? So bache, another important question is, see this, from this to this, from this to this, what is it? What is it? Retitestis, vasa efferentia, epididymis, vas deferens, ejaculatory duct. These are the, yes bache Chandana, these are the accessory ducts. What are they? They are the accessory ducts. Is that clear? What are they? They are the accessory ducts. Is that clear? Yes, everyone. Are you sure? Are you sure? Sure? And this is the root as well. So let me tell you this thing clearly. Seminiferous tubule is not an accessory duct. It is the part of testis. It is basically in the testis. It is not an accessory duct. It is the most common mistake that students used to make. It is not the accessory duct, right? So accessory ducts, they will start from retitestis and from that till that ejaculatory duct. Clear? So this is what you people need to keep in your head. Done, bache? Done. So now we will discuss about the internal structure of the seminiferous tubule. Fine. What we need to discuss? We will discuss the internal structure of the seminiferous tubule, which is very important to understand. Clear? Clear, bache? So let's say you know that 1 to 3 highly coiled seminiferous tubules are there. This is what we have just discussed. 1 to 3 highly coiled seminiferous tubules are there. Let's say you have taken a section of it. Let's say you have taken a section of it, a cross section, right? Let's say you have taken a section of it, a cross section, right? Cross section, right? So now in the seminiferous tubule, see, these are the tubules. Obviously, in the surrounding, of course, there will be the connective tissue, yes or no? There will be the connective tissue. Just imagine within the testis, in the testicular lobule, you have 1 to 3 highly coiled seminiferous tubule. You are going to take a cross section of it. Clear? You are going to take a cross section of it. So obviously you will see the seminiferous tubules there. In the surrounding, there will be the connective tissue, right? There will be the connective tissue. Now, see, look at it, right? Look at it. This is something important, huh? Or you can say that very important. So now here, these coiled structures, okay, now here, these coiled structures are basically your seminiferous tubules, right? And here, in between these seminiferous tubules, in between these seminiferous tubule, in the stroma here, in the connective tissue here, this is your, what is it? It is the Leydig cell. These are the Leydig cells. Important it is, bache. These are the Leydig cells, also known as interstitial cells. What is it? These are the Leydig cells, also known as interstitial cells. Interstitial cells. Basically, they are endocrine cells. They are going to release the male sex hormone. Okay, they are what? They are the endocrine cells that are going to release the male sex hormone. And here what do we have? Seminiferous tubule, right? What do we have here? We have the seminiferous tubule. Clear? Now in the seminiferous tubule, right? Now in this seminiferous tubule, just have a look. This portion, Leydig cell, interstitial cells, they are the endocrine cells. This is the seminiferous tubule. Now here in the seminiferous tubule, the outermost covering will be tunica. It is also tunica. Right, tunica propora. This outermost covering is also the tunica propora. I know it is not given in the NCERT, so many times you do not pay attention on this covering, right? Many times you do not pay attention on this covering, this, this internal structure, So here, this, just a minute, kids. Yes. Right, so now we will firstly finish this seminiferous tubule, its internal structure, then we will move ahead, right? So here you can see these are the Leydig cell, interstitial cell, endocrine cell, which will release the male sex hormone. This is the seminiferous tubule. So here in the seminiferous tubule, obviously there will be some coverings in the surrounding. Like if I talk about the external most covering, as I said, it is tunica. It is tunica propria. Yes, tunica propera. I need to check the spellings. Okay. So, but innermost here is your this covering. This covering is very important. Kindly focus. Okay, this covering here is very important. So can you see I'm making some cube-like cells, some cuboidal cells? Can you see here I'm making some cube-like cells, cuboidal cells? Huh? Can you see this? We are talking about the histology of the seminiferous tubule now. What are we discussing? We are talking about the histology of the seminiferous tubule. So I'm telling you about the covering here. So if you look at it very carefully, these cells, right, this cuboidal epithelium, this cube-like cells, cuboidal epithelium is what? It is the germinal epithelium. What is it? It is the germinal epithelium. But when you talk about the seminiferous tubule, the inner lining, this inner lining of cube-like cells, which is known as germinal epithelium, it is a lining which will further form sperms. Right, this is what we study. It is the lining that will further form sperms. But just look at the structure very carefully. I told you there will be the cube-like cells, the cuboidal epithelium will be there, and that cuboidal epithelium is germinal epithelium. It is a MCQ. So please take a note on it. Take a note of it. And here you guys can also see these cells. Right? Can you see these cells? these columnar cells. Do you know the meaning of these terms? Cuboidal or columnar? Cuboidal means cube-like cells, of course. Columnar, the cells which are tall and cylinder and the nucleus is present at the base. These are the two terms I'm using right now, isn't it? Isn't it? So one is cube-like cell, cuboidal cell, another is tall and cylinder cell. Another one is tall and cylinder cell where nucleus is present at the base. So such type of cells are the columnar cells. So here in the seminiferous tubule, you guys can see this. You guys can see this particular cell. This is the columnar cell. This cell is the Sertoli cell. Sertoli cell. It is also known as sustentacular cell. Right? It is also known as nurse cell. Can you see this? So we are talking about the inner layer of seminiferous tubule. We are saying it is lined with the cuboidal epithelium. We are using the term germinal epithelium for it. And in between in this cuboidal epithelium, you will find some columnar cells. These cells are nothing. They are the Sertoli cells, nurse cells, sustentacular cells. These are the cells which are going to develop the which are going to nourish the developing spermatozoa, right? They are going to nourish the developing spermatozoa. Clear bache? Clear bache? So what will they do? They will nourish the developing spermatozoa. Clear? Clear? And here, see, seminiferous tubule. This is what we are discussing, and the diagram is important, kids. Right? So in the seminiferous tubule, in between here, there will be the lumen, the empty space. Like we have the gut na bache, gut. Gut is lined, like we have the gut na, gut is covered, gut lining. In the gut lining, there is a lumen in between. So lumen is present there. And again, I'm repeating. See, cuboidal epithelium, right? Cuboidal epithelium, and at places, what will you get here? You will get these Sertoli cells, tall and cylinder, right? Tall and cylinder, isn't it? Clear, bache? Clear? The diagram is important. The diagram is important. Do you know why do we call it as germinal epithelium? Thank you so much, Bhavashri. Thank you so much. Do you know why do we call it as the germinal epithelium? Why? Why do we call it as germinal epithelium? Anyone? Anyone? Sanjana? Yes, give it a try. Why do we call it as the germinal epithelium? Quick! Anyone? Anyone? Why do we call this layer, this innermost layer of the seminiferous tubule, as the germinal epithelium? But it's germinal. It will form something. Germinal, right? Means it will form something. Like germination is a normal word, na? I think you all are aware. Like it is going to form something, right? Initially, it was believed. Listen to me completely. Initially, it was believed. Okay, it was believed that this layer used to form the sperms, right? Even if you will read NCERT, you are going to find the same line, that this is the layer which will form the sperms. Like in the NCERT, it is written like that this particular layer, germinal epithelium, is having the cells that are going to form the germ, that are going to form the sperm cells in the future. This is what we study, but actually it is not a fact. Okay, we have to follow the NCERT, whatever is written in NCERT, yes, we have to go as per that, but it is not a fact actually, right? During the developmental stages, what happened, bache? You know, some primordial germ cells. During the embryonic development, what happened? Some primordial germ cells, or you can say that some precursor germ cells. Precursor germ cells. Right, precursor germ cells. You can call them as PGCs. You call it as PGCs, precursor germ cell. Precursor means which are going to form something, right? Right, which are going to form something. So these precursor germ cells, they will, right? Basically, they will find all their way. They will move to the gonads. Basically, from the yolk sac, you will discuss all these terms in the development of the human development basically. So from yolk cells, these precursor germ cells, they find their way towards the gonads. You know na gonads, primary sex organs are known as gonads. Yes, bacho, you know about it, yes or no? Primary sex organs, they are known as gonads. So some precursor germ cells in the initial stages of development, they find their way towards the gonads, primary sex organs, and they used to form the cells which will further form the sperms. Okay, okay. Right, but initially it was believed the lining of seminiferous tubule, it is having, you know, some cells which used to form the sperm. So no doubt we have to proceed it in this way. We have to go like that only, right? But actually this is the fact that from yolk sac, some precursor germ cells, they will find their way towards the gonads, right? And they are the cells, right? They are the cells, these precursor germ cells, which are going to form the sperms. This is the actual information. Clear, bache? This is the actual information. But okay, fine. Still it is described as germinal epithelium, which is incorrect, but we have to follow the NCERT. First point clear? Yes, everyone. First point clear? Now, here, right, as I said, these Sertoli cells, what are they? You will only find them in mammals, also known as nurse cells, also known as sustentacular cells. But they are going to nourish the developing spermatozoa. What will they do? They will nourish the developing spermatozoa. Basically, understand this process. From this layer, right, these germ cells will start behaving like spermatogonia. They used to form the sperms. We will discuss the process today only. Okay, we will discuss this process in detail. We will discuss it today only. So basically what is happening from this layer, right? From this layer, what is happening, bache? The germ cells, right? These germ cells, these spermatogonia, they are going to form the sperm cells, right? So Sertoli cells are going to nourish it. So here the another important topic is the function of Sertoli cell. In the paper, they can ask you the question from this part that what is the function of Sertoli cell? So can you tell me? Yes, bache, can you tell me the function of Sertoli cells? Can you tell me the function of Sertoli cells? Yes, Sertoli cell, obviously the very important function is to nourish the developing spermatozoa, to nourish the developing sperm or spermatozoa, right? And these Sertoli cells, they will form blood test barrier. Do you know that? This is something very important related to it. They will form the basis of blood testis barrier. They will form the basis of blood testis barrier. I will explain all the points right now. Blood testis barrier, and they will phagocyte destroyed sperm. The sperms that are destroyed, they will eat it, right? They will destroy it. Clear? So Sertoli cells, they release androgen binding protein ABP. Androgen binding protein. Clear? Clear? Sertoli cells, they also release inhibin, which is of course a protein hormone. Inhibin and anti-Mullerian hormone. Inhibin and anti-Mullerian hormone. As I said, the function of Sertoli cell, it is very important, right? It is very important. Clear, bache? Clear, bache? This is what you need to write down. And even they form testosterone from estrogen. Okay, so now let me explain their functions. So I think first point is clear to you all, isn't it? I think you know about this first point, right? You people know about this first point, that they are going to provide nourishment to the developing sperm and they form the basis of blood testis barrier. Actually, what is the point here? Just have a look of this diagram, right? Right, have a look of this diagram. Actually, these sperms, they will form here. They will form here, they will grow here. Like let's say this is the seminiferous tubule. This middle part is the lumen of seminiferous tubule. Take the example of a pipe from within. It is hollow. It is not a solid structure. It is hollow. Take the example of a bamboo. inside it is hollow, outside there is a covering. So here in this particular covering, what is happening? Here in this particular covering, there are the cells, right? Here in this wall, basically in the wall of seminiferous tubule, in the wall of seminiferous tubule, the sperm stages will grow. The sperm stages will move, they will grow. Are you getting it? Sperm stages will grow here in the wall of the seminiferous tubule, and as I said, this part is the lumen, right? This space here is the lumen, and in the wall here, what will happen? The sperm stages will grow. This is the first point. Now, obviously, here in this seminiferous tubule, Sertoli cells are also there. As I said, they will provide the nourishment. But what is the story? These sperms are not in the direct contact with our body, right? They are not with the direct contact of human blood. Not like human blood, right? Male human blood cannot recognize the sperm as its own cell. Let me tell you the reason. You know the process of insemination, isn't it? You know the process of insemination. In the last class I have explained you. Yes or no, bache? Do you remember this? In the last class, I told you the first event in reproduction. I gave you the trick, na? Give, sip and go. Give, sip and go. This is what I have given you in the last class. The trick, give, sip and go. Right? So, basically, what is the point, bache? The second step, the second event there, insemination. What is insemination? The transfer of male gamete within the female genital tract. What is insemination? The transfer of male gamete within the female genital tract, right? So this sperm, right, which is a male gamete, on the sperm, you will find some receptors. You will find some receptors which will bind with the ova, right, which are going to bind with the ovum. On the sperm, on the spermatozoa, you are going to find some receptors which will bind with the ovum, isn't it? Isn't it? The structure is like this, bache. So now here, when the sperms they are developing, they have some receptors which will not be recognized by the human body cells, right? In the blood, we have our immune cells, yes or no? This is the basic science. In our blood, you know that whether there is a male or a female, in the blood, immune cells are present. In the blood, what do we have? Immune cells. Immune cells are present. Isn't it? Immune cells are present. Yes or no? Yes or no? Tell me quickly, yes or no? Of course. है ना बच्चे? Of course. They are present. They are present. Right. So if sperm cells, they will come in the contact with the blood. The blood immune cells will not recognize these sperm cells. They will destroy it. Okay. The immune cells present in our blood, in the human blood, they will not recognize the sperm cells and they will destroy it. Okay, so here these Sertoli cells, they form this blood testis barrier. Okay, so sperm cells, they do not get nourishment directly from the blood. Ultimately, it is the blood which is having all the essential things. So sperm cells will not get nutrition directly from the blood. From the Sertoli cells, they will get it. So that is why we are saying that Sertoli cells, right, that is why we are saying that Sertoli cells, they are acting as they are forming a blood testis barrier, right? Blood testis barrier. Now, let me explain it in another way. You know that in the case of, in our case, and especially in the case of males, right, when they attain the puberty, do you know the meaning of this particular word, puberty? Puberty is when reproductive organs, they become functional. Okay, puberty is when reproductive organ, they become functional. So in the case of males, the testis the sperm formation starts at the age of puberty. When they attain the puberty, sperm formation starts. So obviously these cells are not embryonic, right? They are not embryonic, isn't it? They are not embryonic, right? So blood is not able to recognize these sperms. Fine, so they can destroy it. So Sertoli cells, they will form the blood testis barrier. So with the help of Sertoli cells, the sperm, the developing sperms, they will get the nourishment. Okay, they are going to get the nourishment. Like in the seminiferous tubule, that is how Sertoli cells are present. Just check out your NCERT diagrams. Right, check out the NCERT diagrams. This is the Sertoli cells, and this Sertoli cells, because it is having some irregular structure, right? So here you will find different stages of sperm development. So this Sertoli cell will nourish it. Okay, this Sertoli cell will nourish it. So first point clear, second point clear. Now they will phagocyte destroyed sperms or injured sperm. Let's say any sperm which is injured, which is destroyed, it will be engulfed by the Sertoli cells. Clear? It will be engulfed by the Sertoli cell. Now what is this ABP? Androgen binding protein. But in the last class also I have used this word, that is androgen. है ना? In the last class also I have used this word, that is androgen. Androgen means sex hormone here. Male sex hormone. What is the meaning of androgen? It is male sex hormone. So male sex hormone is testosterone. Most active potent form is dihydrotestosterone, isn't it? Dihydrotestosterone. So here this ABP, androgen binding protein, will be released by Sertoli cell to concentrate, right? To concentrate the testosterone. To concentrate the testosterone. Is that clear? Is that clear? Why is it released? To concentrate the testosterone, right? Let me explain this. See. In this diagram, here you can see this is the seminiferous tubule within which your Sertoli cells are present, and here in the surrounding Leydig cells are there. But as I said, I am repeating this again, the Leydig cell, they will release, they are endocrine cell, so they release androgens, they release male sex hormone, they release testosterone, and you know that testosterone is very important, isn't it? All of you know that testosterone is very important, Shamsheer, right? This testosterone is responsible for gamete formation. This testosterone is responsible for secondary sexual characters, like in the case of males, the development of that beards, mustache, that further growth of this larynx, which is known as Adam's apple in the case of male. How is it possible? It is possible with the help of what? Testosterone. It is possible with the help of what? Testosterone. So Leydig cell will release the testosterone. We need, male body need this testosterone for spermatogenesis. So what is going to happen? This Sertoli cell is releasing ABP. Right, this Sertoli cell is releasing ABP, androgen binding protein. So because of this ABP, androgen binding protein, this testosterone will diffuse here in the seminiferous tubule. This testosterone will from the surrounding, it will diffuse here within the seminiferous tubule. So this testosterone will help in the process of spermatogenesis. Are you getting it, Furkana? Yes, that is how it works. Right, it's interesting now. It's simple, interesting, isn't it? It's quite simple, it's quite interesting. Clear, bache? Sure? Sure? Okay. Done. So see, androgen binding protein will be released by Sertoli cells, by which the testosterone will diffuse into the seminiferous tubule. And what is going to happen here, bacho? What is going to happen here? Right. This testosterone will help in the spermatogenesis. This point clear? This point clear? Yes. So even, you know, in the male body also estrogen is present. Are you aware of it? Yes. Bache, are you aware of it that in the male body estrogen is present? Tell me. Do you know that? Do you know that? Quickly tell me. Yes? Anyone? Tell me. Bache, estrogen is present, right? Even in the female body, you will see testosterone, right? Even in the female body, testosterone is present. Okay, just a minute, bache. Done. So see, even in the female body, testosterone is present. Even in the male body, estrogen is present. But their concentration, it varies. Now, in the male's body, this estrogen is very important. Let me tell you. Estrogen provides the motility to the sperm, bache. It provides the motility to the sperm. Right? It is, or you can say that it is very important for the sperm motility, in other words. Right? Important for the motility of sperms. Okay, so if there is no estrogen in the male body, sperms will be immobile. Sperms will be immobile. Are you getting it? So let me tell you, even in the female body, testosterone is there, but the amount is, concentration is less. Okay, because if the concentration of testosterone in female body will be more, hormonal imbalance. If the concentration of estrogen in male body is more, again hormonal imbalance. So it should be balanced, but that doesn't mean that this hormone is not present in the male or in the female body. Okay, so in the male body, estrogen is there, which is important for the sperm motility. No estrogen means sperms will not be able to move, right? Again, it will result in infertility. Clear, bache? So Sertoli cells can even convert testosterone from estrogen. Now, it releases inhibin. This is a very important function here. It releases inhibin and anti-Mullerian hormone. Bache, anti-Mullerian. See, during the development, you will, in detail, obviously it is not the part of your syllabus, but see, during the development, during the course of development, there are two ducts. One is the Wolffian, another is the Mullerian. One is the Wolffian, another is the Mullerian. Mullerian hormone will form the female part. Wolffian, Mullerian ducts will form the female part, right? Like female ducts are made from this Mullerian duct and male ducts are made from Wolffian duct. Male ducts are made from Wolffian duct. So if it is a male body, in the starting it will be clear from the chromosomes. You know about sex determination, isn't it? XY male, XX female. So from that chromosomes, it will be clear that it is the human which is going to develop. It is the male which is going to develop, or it is the female which is going to develop, right? But again, so obviously if it is a male body, so Mullerian duct, it should not be there. It should not be there. So Sertoli cells, they also release anti-Mullerian hormone, which inhibits that anti-Mullerian duct growth, right? It will destroy it. It will stop it basically. Clear? This is the role. And when it comes to the inhibin, which is a protein hormone, right? Inhibin, the role of inhibin in the Sertoli cells is associated with the FSH, that is your follicle stimulating hormone. How is it related? It is related with your FSH, that is your follicle stimulating hormone. Basically, right, this inhibin inhibits the secretion of FSH. So obviously, negative feedback will be provided by this inhibin to stop FSH secretion, to stop follicle stimulating secretion. Clear, bache? So this is important. Clear? This is important. And bacho, we will, let me finish the parts of male reproductive system. Today itself, we will start the spermatogenesis. Then in detail, I will explain you the role of FSH and will relate it with inhibin. Okay? Okay? And remember, inhibin is also present in female body. Inhibin in females, you can mention it in your notes. In females, is released by granulosa cells of ovary, right? Ovarian cells used to release inhibin. So inhibin also is present there in the female body, right? It is also present in the female body. Clear, bache? Clear? Sure? Sure? Done? Okay, so any doubt? Done? So we are done with it. So this is the lumen area. This is the lumen area. Clear, bache? So the position of these cells should be clear. So interstitial cell is present in between the seminiferous tubules. Okay, so let me show you the diagram. So have a look. Done. So this part, the internal structure clear. So you know that retitestis, from retitestis vasa efferentia will arise, and then here they are going to form this epididymis. Clear, bache? Now we will discuss this epididymis. right? So this is basically the epididymis, this part. Can you see this? This part. This part is the epididymis, and further this epididymis will further form this vas deferens. Clear bache? Clear bache? So this epididymis is present on upper dorsal or posterior part of the sperm, right? This epididymis is present on this upper, okay, upper dorsal or posterior part, epididymis. Now, bache, whenever we talk about epididymis, I always mention this. It is the part where there is the temporary storage. Focus on this word, huh? Temporary storage and maturation of sperms, right? There is the temporary storage and maturation of sperms. Storage and maturation. Obviously sperms, right? Sperm production will start when the boys will attain the age of puberty. Sperm production will start when boys, they will attain the age of puberty, right? And it will be for the lifetime. With age, of course, it can happen that with age, you know, the quantity decreases, but the sperm production will not stop. In the case of boys, it will keep going, but with the age, you know that aging is the factor, otherwise it will be there. Okay, now sperm production is too much. In the case of you know that in nature male gametes are too much. Okay, so whenever the sperms form, the temporary storage is there in this epididymis. As I said, in the epididymis, sperms can remain stored for even for a month. And here also there will be the maturation of sperms. This is very important. This is very important. You know, sperms will produce here in seminiferous tubule. Seminiferous tubule is the actual site of sperm production. But after their production, they should get stored in epididymis. Let's say, right, we are directly taking out sperms from the seminiferous tubule. Just take one example. In a human male body, sperms are forming here in the seminiferous tubule. We are directly taking that sperms, right? They are not getting a chance to get stored here in the epididymis. So you will find, bache, that these sperms are not movable. Okay, they are not movable. They are not mature enough. They are not capable enough to fertilize the female gamete. Are you getting it? So their storage is very important in epididymis. Their storage is very, very, very important in epididymis. Clear? Clear? So after the sperm production, at least for 18 hours to 24 hours, they should get stored here in epididymis. They should get stored here in epididymis. Clear, bache? So maturation of sperm will be there in epididymis. Fine. So although it is not given in NCERT, but let me tell you, there are three parts of this epididymis: head region, caput, epididymis. This is the head part, right? See, this is how I remember. Head, right? On our head we used to put a cap, na? Cap. So caput, epididymis, the head region. Then comes the corpus, right? Then comes the corpus, epididymis. Corpus means body, right? And when it is the posterior end, tail end, it is when it is the tail end, it is the caudal epididymis, right? It is the caudal epididymis. So in three parts we discuss it: caput, corpus, caudal. Caput, corpus, caudal. So caput is upper highly coiled part. This is the middle part, and here it is the basal or the least coiled part. Clear, bache? Clear? Yes. Done. So now this epididymis, as I said, it is going to enter in the abdominal cavity, right? This epididymis will enter in the abdominal cavity. Let me tell you, this coda, the basal part of epididymis, enters abdominal cavity. It enters abdominal cavity. Clear, bache? Right, it enters abdominal cavity in the form of vas deferens. Clear? So basically in the scrotum, you know that it is present, then vas deferens will form, then obviously it will leave the scrotum, it will further enter in the abdominal cavity, then this vas deferens from both the testes, it will, as I said, it will coil near to the ureter, it will coil over the urinary bladder, then finally will open up in the urethra. So if there is any doubt from this part, please let me know, kids. If there is any part, please let me know. Quick. Quick, quick, quick. Done? Done, bache? So now, bache, we need to talk about the glands. See, look at this diagram. Can you see this? See, vast difference from both the sides. So it will coil near ureter. It will coil over this urinary bladder. Right, bache? Right. So here you guys can see. So now, in the case of male reproductive system, we will also talk about the accessory glands. What are we going to discuss in the case of male reproductive system? We will talk about the accessory glands. Are you aware of it? So here we have pair of seminal vesicle. What do we have? Here we have pair of seminal vesicle. We have a prostate gland, pair of seminal vesicle, prostate gland, which is single. It is not paired. It is single, but it is lobed, right? And one is the pair of bulbourethral gland, also known as Copper's gland, right? It is the bulbourethral gland, also known as the Copper's gland. Are you getting this? Yes, bache, are you getting this? So as I said, what do we have here? We have three accessory glands in the male reproductive system, right? We have the pair of seminal vesicle, prostate gland, and pair of bulbourethral gland. Pair of seminal vesicle, prostate gland, pair of bulbourethral gland. And look at their position. Seminal vesicle, this is the prostate, although it is single but having many lobes, and these P-shaped bulbourethral gland. Clear, bache? Clear, bache? Okay. See this. I think this diagram can clear it. See this. Testis. This is the epididymis. This is the vas deferens. Look at this vas deferens. Right, right. And here it will coil somewhere here, this vas deferens. And see this part. It is the seminal vesicle. Right, this is the prostate gland. So prostate gland will open here in urethra, which will be there in penis, and then bulbourethral gland. So any doubt from this diagram? Any doubt from this diagram? Yes, everyone, tell me quickly. Quickly, see this. Right, diagrams are literally amazing. Seminal, prostate, and bulbourethral. And see, they all will open here in penis, which is having the urethra. Right, which is having the urethra. Done? Sure? Okay? Sure, bache? Okay. So now let's talk about it. First of all, understand this. This is one vas deferens. This is another vas deferens. You will see it will also form some, widened structure ampulla. But right now, I need to highlight something else. See this. This diagram is important to understand, bache. Can you see this? Yes, can you see this? So ultimately, what is the point here, bache? See, this is the vas deferens, as I said, and here you have the pair of seminal vesicle. Right, here you have the pair of seminal vesicle. Now, if you will observe, this vas deferens is receiving the secretions from seminal vesicle. Seminal vesicle is a gland. Gland. What is the meaning of a gland? Gland is something which is going to secrete, right, which will provide us the secretion. And of course, that secretions are used for the maturation of sperm. They are important for the maturation of sperm. They are important for the sperm function. Isn't it? Isn't it? So when this vas deferens, it receives the secretion from the seminal vesicle, together they are making what? Together they are making the ejaculatory duct. Right? Together they are making what? Ejaculatory duct. So basically the red color that I formed here is, right, the red color duct that I have formed here is ejaculatory duct. Which one? Which duct is it? It is the ejaculatory duct. So ejaculatory duct is formed when vas deferens, it receives the duct of seminal vesicle. Clear? Right? And finally here the prostate gland, it will also pass its secretion to the urethra. So ultimately they all will open up in the urethra. So urethra is basically present where? It is present in the penis. Right? It is present in the penis. Is that clear? Where is it present? It is present in the penis. And here nearby also you will see the bulbourethral gland and its secretion. Done, bache? So first of all, let's focus on the secretion of this seminal vesicle. So what do you know about the seminal vesicle other than the fact that it is a paired gland? Yes, what do you know about the seminal vesicle? Yes, bache, what do you know about the seminal vesicle? Quick. Seminal vesicle, it is lubricant. It is lubricant, jelly-like part, and it forms 60 to 70% of semen. What will it do? It forms 60 to 70% of semen. So maximum contribution of semen is from this seminal vesicle. 60 to 70% of semen is formed by this seminal vesicle. Obviously it's a gland, right? It's a gland, it will secrete something, isn't it? It's a gland, it will secrete something. So lubricant, jelly-like part which forms 60 to 70% of the semen. So obviously it is alkaline. The pH is near about 7.3. Right, and if I talk about its secretion, remember the secretion of seminal vesicle, they provide energy to the sperms. Right, the secretion of seminal vesicle, they provide energy to the sperm, because here in the fluid of seminal vesicle, fructose is present. Right, fructose is present in fluid of seminal vesicle. Clear, bache? This is what you need to focus. Fine. So basically sperms, they don't take their energy from glucose, they take their energy from fructose. Okay, they take their energy from fructose. So whenever there is any test for the rape now, right, so although there are many, and with the time, you know, the medical examination, the things in the medical examinations have changed, right? But the point here is, this is what you need to focus, like whenever there is a sexual intercourse, so ultimately in female body there is no chance of presence of fructose in the vagina, right? There is no chance of presence of fructose in the vagina. So if there is the sexual intercourse, definitely, right? If there is the unprotected sexual intercourse, definitely the fructose will be there in the female vagina. So that confirms the sexual intercourse. So during rape test, you know, these are the while checking whether there is a rape or not, right? So they do different type of medical examination in which they collect the swab, they collect the fluid, they collect the fluid from the female body. So this is what they used to check. Okay, this is what they used to check. So when you are talking about the seminal vesicle, as I said, alkaline it is, fructose is present in the fluid of the seminal vesicle. Clear, bache? Clear, bache? So other than that, some, you know, clotting agents like your fibrogen, prostaglandins, which are meant for the contraction of, right, which are meant for the contraction of female reproductive system, prostaglandins, right? Other than that, which is some citrates, inositols, some enzymes are present. Clear? Some enzymes are present. So they can ask you two questions from this part. Number one, right, which gland provides the maximum, which gland is having the maximum contribution in the semen? That gland is what? That gland is your seminal vesicle. That gland is what? That gland is your seminal vesicle. Secondly, which gland will provide the fuel for the sperm? That fuel, F for fuel, F for fructose, that is by a seminal vesicle. So in the seminal vesicle secretion, fibrinogen, right, fibrinogen your prostaglandins, which are responsible for the contraction of the female reproductive tract, right? Citrate, inositol, right? And other proteins are present. So this is the secretion of seminal vesicle. So secretion of seminal vesicle is meant for providing the energy. It provides what? Energy to the sperm. Now the next is bache, prostate gland, which is single but having many lobes, and it will provide its secretion to the, it will release its secretion in the urethra, right? Prostate gland. Again, its secretions are also sticky. They are also lubricant, right? These secretions are also sticky. They are also lubricant, right, bache? So basically, it is going to secrete alkaline fluid, right? Which it is going to secrete alkaline milky fluid, right? It is again alkaline. Alkaline means basic it is. So alkaline milky fluid bache, alkaline milky fluid bache, and it forms 30% of the semen, right? So it helps in the activation of sperm. Its secretions are playing role in the activation of sperm. Remember this. This is important. So as I said, secrete alkaline milky fluid, which is sticky or jelly-like. This is what you can write, sticky or jelly-like. And 30% of the semen, it helps in the activation of the sperm. And moreover, bache, the composition of this prostate gland, right? It helps in the clotting of the semen. Do you know that? It helps in the clotting of the semen. Like when the secretion of prostate glands and the seminal vesicle, it get mixed. See, now you know it very well that seminal vesicle, the duct of seminal vesicle will receive the vas deferens, right? So obviously when sperms and the secretion of seminal vesicle, when they will get mixed, right, the semen will get coagulated. It will get clotted, because here in the prostate gland, we have some clotting agents that will clot the semen. Why? Because when semen is clotted, the sperm motility will decrease. Sperm will be able to save its energy, right? Because that energy is required in the female genital tract. Okay, so here what is going to happen, bache? Here what is going to happen? The prostate gland secretion, they are going to coagulate the semen. Clear, bache? Clear? Yes or no? Yes or no? So here also you will find the citric acid in the secretion. You will find citric acid in the secretion. Calcium ions will be there in the secretion. Some clotting enzymes are present in the secretion, right? Some pro-fibrolysin enzymes, right? They are present here in the secretions of the prostate gland. So composition is important, right? Composition is important. Clear, bache? Done, bache? Yes, right? So again pro-fibro, it's fibrino. Clear? So like here in the structure of this seminal vesicle, here in the composition of seminal vesicle, we can see fibrinogens, clotting agents are present, right? So see some enzymes, pro-fibrinolysin enzymes are also present. They will get activated later. Okay. So this is what you people need to keep in your head. Done. So when the secretions of, I'm writing this point here, when the secretions of prostate gland, when the secretion of prostate glands get mixed with secretions of seminal vesicle, right, the semen gets clotted, right? Semen gets clotted. And when semen gets clotted, what will happen? Sperm motility decreases. Sperm will store, the energy will be saved. Okay, sperm will become less active. Clear, bache? Clear? Done. Energy will be conserved. So later on, these enzymes, fibrinolysin and all, they will again liquefy the semen, and again, the sperms will become active. Done. So bache, last gland which is mainly for the lubrication of penis is the Copper's gland. Okay, it is the Copper's gland. What is it? It is the Copper's gland, also known as it is bulbourethral gland. What is it? It is bulbourethral gland. So its secretions are going to lubricate the penis. One thing, right? The main role is the lubrication of penis during sexual intercourse. During sexual intercourse, what will it do? Yes, everyone, just type it in the chat section. Just type it in the chat section. Guys, see, those who are, sending the messages here, right, Ghazal and all, they are not serious NEET aspirants, of course, right? They are just wasting your and my time here. So ignore them. Do not revert. Okay, do not revert. And if any student is a partial dropper and if still that student is not sincere, so you know that. There is no need to entertain such students. So focus here. Fine. So what is the role of this Copper's gland? Lubrication of penis during sexual intercourse. It is the important role, and basically it reduces the acidity of the urethra. What will it do? It reduces the acidity of the urethra. Clear, bache? Clear, bache? Clear? Yes. So its secretions are also jelly-like. It is also acidic. It reduces or destroys the acidity of urethra. Okay? Okay? Slightly alkaline secretions are there. Slightly alkaline, like you can say that pH is near about 7.2. So it destroys the acidity of urethra for easy movement for clearing the path for the sperms, basically. Done, bache. So lubrication of penis is the main role. So secretions are, of course, jelly-like. Done. So maximum contribution is of seminal vesicle. Clear, bache? Maximum contribution is of seminal vesicle. Is that clear? So any doubt? Yes, bacho, any doubt? Any doubt? Done? Sure. So again, let's have a look of this diagram. I think everything is clear, crystal clear to you. Look at the position. So see, finally everything, see, they will open up in the urethra here. And where is this urethra present? Where is this urethra present? In the male external genitalia, in the male copulatory organ, that is penis. Right? In the male copulatory organ, that is penis. Clear, bache? Clear? So now this is what we need to discuss. Then we are done with the male reproductive system, and then we will talk about the spermatogenesis. Okay. Now, penis. As I said, it is the male external genitalia. It is the copulatory organ. What is it? It is the male external genitalia. It's the male copulatory organ which will help in, yes, it is the male copulatory organ which will help in what? Which will help in help during sexual intercourse. So, bache, penis is made up of, it is made up of special erectile tissue, right? It is made up of special erectile tissue. Fine, erectile tissue. Actually, what is happening? Penis is muscular, and here in the penis, there will be the blood flow, right? There will be the blood flow. That blood flow is responsible for the erection of penis. During sexual intercourse, during sexual arousal, blood will flow towards the penis, and because the erectile tissue is present there, the penis will become erect. So penis is muscular as well, and it is having the erectile tissue. So now in the case of penis, if I talk about the diagram, now you know that erectile tissue is there. So in the penis here, if you talk about this tip here, this tip part, this is a little bulging. Right, this is bulging. So we call this part as glans penis. Right, it's the highly sensitive part of the male external genitalia, glans penis. Right, it is the glans penis. You need to know about it. Now, this glans penis is covered with the skin. Right, with a loose fold of skin, it is covered. Clear? It is covered with what? It is covered with the loose fold of skin. Loose fold of skin, and that is known as prepuce or foreskin. That is known as prepuce or foreskin. Clear, bache? Clear? Now in the case of male system, obviously here there will be an opening. In the case of males, this opening is common for the semen as well as for the urine. Clear, bache? This opening, for opening we also use the word meatus, right? So it is the urinogenital meatus or urinogenital aperture. It is common, right, bache? It is common for both. It is common for urine as well. It is common for the semen. So when semen will come out, urine will not come out. When urine will come out, semen will not come out. Okay? So we also use the word meatus for it. Meatus, M-E-A-T-U-S, opening, right? Meatus means opening. Okay. Done? Clear, bache? Sure? Sure. So erectile tissue it is. So there will be the blood flow during the sexual arousal, right? So there are 3 erectile tissue, 3 bundles of erectile tissues are there. 3 bundles. So one is your corpora cavernosa, corpora cavernosa, and corpus spongiosum. So it is not given in NCERT, but once there was a question in the AIMS examination. So corpora cavernosa, right, bache? Corpora cavernosa and corpus spongiosum is there. Okay, corpus spongiosum is there. So what are they? These are the erectile tissue, right and left erectile tissue, corpora cavernosa, corpus spongiosum, right? So corpora cavernosa 2 in number, 3 in number. So these are 3 longitudinal bundles. Done, bache? So it is present on the right side, corpora cavernosa, corpus spongiosum. So bache, corpora cavernosa will not reach till the end. Okay, it will not reach till the end, till the tip. Right, it's a kind of rough diagram. And one will be your actually it is not like this. On the right side you will be having corpora cavernosa, on one side there will be the corpus spongiosum. So corpus spongiosum will reach till the tip, but this corpora cavernosa, it will not reach the end of the penis. Okay, it will not reach the end of the penis. Fine? Done? Done, bache? Sure? Done? So that is how it is present. Fine? So if you will see the section, so you will find it like this. So these 2 are corpora cavernosa and this one is corpus spongiosum. Okay, so ultimately during sexual arousal, as I said, blood flow will be there. So bache, it is controlled by autonomic nervous system. This blood flow, this vascular part, basically it is controlled by the autonomic nervous system. Okay, it is controlled by the autonomic nervous system. I'm telling you this thing. Erection of penis because of blood flow is controlled by autonomic nervous system. Do you know this? Autonomic nervous system, central nervous system, autonomic nervous system. Do you have any idea? So can you tell me what will be there in the autonomic nervous system? Anyone in the class? What will you see there in autonomic nervous system? Can you tell me? Yes, bacho, can you tell me what will you see here in the autonomic nervous system? So tell me, then I'll explain this diagram. Okay? Quick. So I think now everything is right. Everything is control. Everything is discussed. Okay. Okay. So if still there is any doubt, do let me know. Yes, if there is any doubt, kids, please tell me. I'll check if there is anything. else that I need to tell you? No, nothing. We have already covered it. But can you tell me the word for the removal of testes? If there is the removal of testes, which word do we use for the removal of testes? Anyone in the class? Yes, do you know the word for the removal of testes? Very good, Priyanka bache. Very good, Priyanka. Do you know the word for the removal of testes? Yes, let's complete this part, then there will be the final revision. Anyone in the class, what is the word that we use for the removal of testes? Bache, that's castration, right? That's castration. Castration means removal of testes. So obviously if testes are not present, sperms are not there, that person will be infertile, right? That person will be infertile. Castration, removal of testes. Okay, now there is one more word. I am telling you now that it is the ANS. It is the ANS which will regulate the erection of penis. Erection of penis during sexual intercourse. And I asked you what will be there in ANS. So ANS will include your parasympathetic nerve fibers, right? ANS will include your parasympathetic nerve fibers and your sympathetic nerve fibers. Fine. Parasympathetic nerve fibers and your sympathetic nerve fibers. So one is responsible for the erection of penis, another is responsible for the ejaculation of semen. Clear, bache? Like your this parasympathetic is responsible for the erection of penis, and this one sympathetic is responsible for the ejaculation of semen, right? It is for the ejaculation of semen. Clear? Clear? And now see, if you talk about the normal sperm count, so it is 20 to 120 million per ml. Do you know that? It is 20 to 120 million per ml. 20 to 120 million per ml. Now let's say if it is less than, let's say if it is less than 20 million per ml, so it will be oligospermia, right? It will be oligospermia, isn't it? It will be oligospermia. If in the semen sperms are absent, then it will be zoospermia, right? Then it will then it will be azoospermia. Fine, it will be azoospermia. Clear, bache? So castration is the removal of testes. ANS includes sympathetic and parasympathetic nerve fibers, right? Normal sperm count is 20 to 120 million per ml. Oligospermia when it is less than 20 million per ml, right? Azoospermia when there is the absence of sperms, right? When there is the absence of sperm, that is the azoospermia. So these are the terms which are important. And during single ejaculation, a male can ejaculate 200 to 300 million sperms. Just imagine, in single ejaculation, right? If the male is healthy, in single ejaculation, male can release 200 to 300 million sperms. Clear, bache? Clear? During single ejaculation, during coitus. Okay? Done? Done? Clear? So normal sperm count, this less than 20 million per ml. Oligospermia, sperms where sperms are semen where sperms are absent. Azoospermia, 200 to 300 million sperms are present in single ejaculation in each coitus. So out of it, 60% will have normal shape and size, 40% will show vigorous activity, right? Vigorous motility, vigorously they will move. Clear, bache? I'm repeating this point again, right? So 200 to 300 million sperms are present in single ejaculation, right? And out of it, 60% should have normal shape and size, 40% should show vigorous motility. Fine, bache? It should show vigorous motility. So that is what you need to take care. Done, bache? Done. So these terms are important. Okay, so write down, make the notes, bache, do not miss these points. So this is all about the male reproductive system. Clear, bache? This is all about the male reproductive system. So any doubt? Tell me now, do you have any doubt? Bache, I don't know. I have to check, Soumyadeep. Done. So if there is any doubt related to this topic, please ask me, bache. And next that we need to discuss is your spermatogenesis. So see, you can even check it from NCERT. Right, bache, we have covered everything from the NCERT part. So see, the scrotum, right, it is outside the body, so that is why testes are extra abdominal. They helps in maintaining the low temperature of testes, that is 2 to 2.5 degrees Celsius, normal, lower than the normal internal body temperature. Clear, bache? So in adult, testes are oval in shape with a length of about 4 to 5 centimeter, width of 2 to 3 centimeter. So please read it from NCERT, even note down the dimensions in your notes, bache, because it is given in NCERT, they can ask it. Chances are less, but still such type of questions can come in your paper. So testis is covered by dense covering. You know about the covering, right? 250 compartments are there and we call them as testicular lobules. So testicular lobules are covered with, Chandana. Testicular lobules are covered with, yes, testicular lobules, they are covered with, anyone in the class? They are covered with testicular lobules, they are covered with, anyone? Tunica vasculosa, right? They are covered with tunica vasculosa. Clear, bache? They are covered with tunica vasculosa. None. So see, male germ cells, Sertoli cells, the function of Sertoli cells, Leydig cell, interstitial cells, we have covered everything. And yes, mention one more point, bache. Leydig cells synthesize and secrete testicular hormones called androgens. Other immunological competent cells are also present. Right? Here in the seminiferous tubule, in this part, we will see some other immunological competent cells. Okay, some other immunological competent cells are also present. Okay, done, bache. So bache, PDF will be shared in the Telegram group, because right after your class I have another class in my batch. Okay, so I will share the PDF, and tomorrow we will increase the speed. Okay, so we will cover the female reproductive system and we will try to finish the gametogenesis there. Okay, so in the morning, because right after your class I used to have class in my Avengers batch, so from coming days we will keep that schedule 10 to 11:45, right? We will fix it 10 to 11:45. Okay, so this week I'll keep the class daily so that we can finish the human reproduction chapter in this week, and then we will revise it. Okay, then we will revise it. Okay, without spectacles you are looking like with spectacles. With spectacles, Soumyadeep, I don't look good. This is what you are trying to say. This is what you are trying to say. And don't worry, we are going to increase the pace as well. Okay? So if there is any doubt, do let me know. And guys, I requested it yesterday, please put a comment. Right? So many students are there in this particular batch. So please put the comment in the comment section also. You like the appreciation, isn't it? Isn't it? You all like the appreciation. Right? I'm not asking for appreciation. I'm asking for the genuine comments. Even if there is any suggestion, if you feel, ma'am, you're not teaching properly, write it in the comment section. But put something there. Ma'am, what about hernia disease in males? Hernia mostly in the case of males, but you cannot say that it is not. It is only in the case of males. Females are also having hernia, but chances are more in the case of males. I'll discuss it in the next class for Kana. Okay, anything else? Now screen is not tilted, it's all good, it's all fine. And as I said, bache, again, you guys can be the part of our energized batch, or need energized batch, or need Avengers batch, whatever you want to say. So you just need to click on the link, right? Add your number here, right? You can continue with the email. So after applying this coupon code, although it is already applied, you will get this batch at a price of 15,000 rupees. But still, this batch is still in a, you can say that you are buying it at a very affordable price. Initially it was for the 10,000 rupees, but it's okay, it's fine. At that time we requested our company, but now we cannot provide you that thing. So now it is for 15,000. Maybe in coming future the price increases. But all I want to suggest is, if you want to join the batch, please join it now. Otherwise, backlogs will be there and you will miss many things. Again, the pressure will be there. Okay. Still you have the time. It will become very difficult if you will start in September or in October. I'm not saying if you will start from the month of September or October, you will not be able to crack it. You can. But again, the chances will be less then. And you have to work very hard. Fine. So take care of these things, and thank you so much for watching.